Good morning, good afternoon, and good evening to everyone. I'm Dr. Jessica Justman, and our ICAP Grand Rounds will be starting in just another minute or two. We'll give time for a few more participants to join. So please stay with us. Thank you. Okay, well, I'm sure we'll have people joining us as we start up. So again, I'm Dr. Jessica Justman, Senior Technical Director here at ICAP, and I want to welcome everybody to today's ICAP Grand Rounds webinar. We are going to be talking about H5N1, bird flu. Certainly, this is something that has been in the news lately. It's an infection that's been widespread in wild birds worldwide and is causing outbreaks in poultry and dairy cows. And there have been recent human cases among U.S. dairy and poultry workers. And the recent increase in these cases have focused more attention on the topic of H5N1 as the threat to human health and the risk of another global pandemic and intensifies. Certainly, the conversations around this has intensified. So we're very fortunate today to have two really outstanding experts who are going to speak to us and provide updates on the global status of H5N1, including the evolving epidemiology of the virus, followed by a situation update on H5N1 in the US and the ongoing response efforts of the CDC. Our first speaker will be Dr. Aspen Hammond from the World Health Organization. Dr. Hammond is trained as a veterinarian and as a public health specialist. She's been with WHO since 2012 and has worked in WHO's global influenza program since 2015. And her responsibilities focus on surveillance and risk assessment of zoonotic influenza, supporting the rapid detection and response to outbreaks and risk reduction efforts of WHO and its member states. Dr. Hammond also works on assessing global influenza epidemiologic and virologic surveillance data and developing guidance on influenza and other respiratory virus surveillance methods and standards. And she also supports implementation of guidance and tools for WHO member states. So we'll hear from Dr. Hammond first, and then I will be introducing Dr. Dugan after for our second talk. Dr. Hammond. And sorry, let me, while we're switching, just encourage everybody, if you have questions for our speakers as we go along, please use the Q&A box for your questions. That's really going to be the best place to put in your questions for the speakers. Thank you. Dr. Hammond.
Okay, thank you. Please just let me know if the screen sharing is not showing the slide set.
It looks perfect. It's great.
Okay. Thank you. Okay, it's a pleasure to be here, and thank you to the organizers for giving me the opportunity to participate. I'm going to speak on the global overview of avian influenza H5, giving a bit of the global context and some information on WHO activities and resources and recommendations, and to be followed by again a bit more in-depth situation in the US. By my colleagues. So first, I just wanted to just to set the scene, make sure we're on the same page together, giving a bit of the background of influenza A viruses, the topic of today. So influenza A viruses are categorized based on the subtypes of their surface proteins, so the hemagglutinin and neuraminidase. So for example, our current seasonal influenza viruses are H5N1 and H3N2. But the focus of today's talk is on animal influenza viruses, for which birds are the natural reservoir for influenza A viruses. For influenza A viruses, there's a wide host range. There are influenza A viruses that are specific to swine, that circulate in horses, dogs, cats, many different kinds of animal species. What's important for today's talk is to understand that sometimes these influenza viruses can infect other species than their usual host. And that influenza A viruses are capable of causing epidemics or seasonal epidemics, but also pandemics. So just to again give some context. So again, the animal influenza A viruses are those viruses that circulate in animals. This includes the avian and the swine influenza viruses. For example, H5 and H7 avian influenza viruses. And for avian influenza viruses, these can be high or low pathogenicity in birds. It's important to note this only refers to the severity in birds and not people. And zoonotic influenza viruses when that animal influenza virus crosses species and infects a human. And pandemic influenza are those animal influenza viruses or novel influenza viruses that may begin to spread among humans. So spreading from human to human, and where that human population has little or no underlying natural immunity to these new or animal influenza viruses. And again, the seasonal influenza viruses are those that are adapted to transmitting from human to human, that cause the seasonal epidemics, and for which there's a vaccination program, and a vaccine that's updated every year for the prevention and control of seasonal influenza. So, given this diversity in different types of influenza viruses, there's a need for a global approach to monitoring all the different circulating influenza viruses for prevention and control, to detecting and responding to novel and pandemic influenza, and especially to monitor that interface between humans and animals, where influenza viruses can cross over from animals to humans. And so I think that the focus of today is looking at the pandemic potential. So just to refresh the necessary factors for a pandemic. So one is there's a new influenza virus, be it an animal influenza virus or a new virus that emerges, starts to cause infections in humans, but also that this virus acquires the ability to transmit from one human to another. And again, a pandemic influenza virus is a virus also that the human population has little or no underlying immunity to. And it's clear that it's changes in amino acids of the viruses that change their genetic makeup or their phenotype, and that there are small changes that can happen that can change how these influenza viruses act. So I think it's important to understand that there are some of these conditions that need to be fulfilled before there is a pandemic. And so going back to the need for a global approach to influenza surveillance, this has been a recognized need for quite a while. So for over 70 years, there's been a global influenza surveillance and response system we call GISRS. That's been the foundation for influenza surveillance, preparedness, and response. The success and the sustainability of this global system is really due to the enormous commitment from the WHO member states and from the support from other international agencies and our partners. And so the map on the screen shows the location of national influenza centers, for which there are around 150. There are also collaborating collaborating centers on influenza, which are specialized centers which can do further characterization of influenza viruses. There are essential regulatory labs which handle some of the regulatory aspects around the influenza vaccine and testing reagents and diagnostics. There are H5 reference labs, and all these labs contribute to the functioning of this global influenza surveillance system. And so if we look at the incidence of human infections with animal influenza viruses or zoonotic influenza viruses, WHO has been monitoring this for quite a number of years. I'm showing here the epidemic curve of human cases that were reported to WHO that we're aware of since 2003. So you can see that for over 20 years, these viruses have been causing human infections. And number two, that there's quite a variety of influenza A subtypes that have infected humans. So the focus of today is H5, but I just wanted to acknowledge there are many other influenza A viruses coming from animals that have infected people. This includes avian influenza viruses, but also swine influenza viruses. They are also occasionally detected in humans that have close contact with swine, where these viruses may be circulating, and humans can acquire the infection from contact with infected animals. So, so again, we can see quite a number of subtypes and continued these sporadic detections of human cases. In the middle, you can see a few peaks of blue those represent different waves of H5N1 virus infections in humans that were detected in China between 2013 and 2017. But no human case with that subtype had been detected in many years. And here, this is the same curve, but only highlighting those cases, human cases of detection of H5 viruses. So we can see we have detections of H5N1, which have happened again since 2003 when the virus reemerged. But also since then, we've had detections of H5N2, of which there was one. We've had some detections of H5N6 viruses, as well as H5N8 in humans. A little bit of background on the H5 viruses, just to emphasize the continuing evolution of these viruses. So the H5 viruses were first detected in 1996, this specific lineage, and have continued to diversify since then. There have been 10 clades, and there have really been only a few of these that have survived to be detected since the emergence in 1996. So the clades that we call 2.2, 2.3.2, and 2.3.4.4 have caused most of the global epidemics in wild birds and poultry in recent years. And from 2016 onwards, it's really two clades that have persisted, and these continue to diversify into multiple subclades. So we use the word clade to be able to describe these groups of viruses that are similar genetically. And it's this clade 2.3.4.4 of H5 viruses that emerged in 2008 and contained certain mutations in one of the surface proteins. It has also, these viruses also reassort with other viruses, creating different H5 subtype viruses. So in the middle is the phylogenetic tree, just showing the evolution of H5 viruses over time. And similarly, the detections by clade are shown in the graphs, the images that are on the slide on the right, showing the continued clade detections and subtype detections over time. These are from animal outbreaks where these viruses are detected. So two points here that I wanted to emphasize regarding these H5 clade 2, 3, 2.3, 0.4, 0.4b viruses, which is a subclade of 2.3.4.4 H5 viruses. One is that since 2020, these clade viruses have spread to many parts of the world, many parts of Africa, Asia, Europe, and North and South America, and even Antarctica. The second thing is, in addition to the geographic spread, is that there have been more species where these viruses have been detected in, and this includes wild birds and terrestrial and marine mammals, especially if you look at the from 2003 to 2019, compared to the most recent 4 years or so, there's quite an increase in the number of animal species from which these viruses have been detected. So two things that are of interest in this particular group of viruses. A critical role of WHO is in risk assessment, and it's important that we continue to monitor these viruses and that we look at the public health risk of these viruses. We do this in a couple different ways. One way is we look at the risk of further human infections. We look at the risk of human to human transmission, and we do this also with our animal colleagues in a One Health approach. So we recently updated our situation assessment with FAO and WOA. So FAO, the Food and Agriculture Organization of the UN, and WOA, the World Organization for Animal Health. So we updated the overall global public health risk, assessing that the risk of the recent circulating H5N1 viruses, the public health risk to be low. There's a slightly higher risk of infection in people who may have occupational exposure, that we expect further infections to happen, but so far there has been no human-to-human transmission. And I just wanted to emphasize that these ongoing risk assessments that we do are reliant on strong surveillance systems, both in the human population and also in animals, the timely detection and sharing of information. And sharing of information includes both the genetic sequence data, which is important, but also the viruses themselves with collaborating centers who can do further characterization and studies on these viruses. Another method for risk assessment that we do is also looking at the specific virus. So there's WHO has a tool for influenza pandemic risk assessment. Again, it focuses on certain viruses with this potential to cause a pandemic. It uses, again, a One Health approach. And this looks at ways to support preparedness for potential influenza pandemics. It also guides research, and it guides the selection and generation of candidate vaccine viruses in case they were needed if there was to be a pandemic. So this risk assessment looks at different characteristics of the virus, its receptor binding properties, the characteristics of its genome, how widespread the infection is in animals, etc. So just two examples of some of the risk assessment activities that WHO does regarding these viruses. Another activity I wanted to highlight of WHO is to look at the development of candidate vaccine viruses. Again, this is for preparedness purposes, but WHO works very closely with the animal health organizations to look at the viruses that are circulating in animal populations, the viruses that are detected in human cases of infection with influenza A viruses that are of animal origin. There are meetings that happen twice a year, with two goals. One is to update the seasonal influenza vaccine, but one is to update the viruses that may be used in case there was a need to develop vaccines quickly. And selecting these viruses ahead of time can make the production of pandemic vaccines happen in a faster manner. We can advance the process for getting the right vaccine, the right virus into the vaccine by doing these preparedness activities. So in the last meeting was in February. I've given the link of the outcome of that meeting, and Also, given the H5N1 detections in the US, there was an update on looking at the circulating viruses and comparing them to the existing candidate vaccine viruses. And we published that report also on our website in May. So the links are there that maybe if you want more information later, you can have a look. The next thing I wanted to turn to is some important recommendations and resources for countries coming from the global level. And one is on surveillance. It's, the objectives of surveillance as we see it at the global level is we want to be able to have countries have the ability to rapidly detect human cases of H5 virus infection. It's important to be able to assess and monitor changes in how these viruses spread to humans. And also we want to be able to detect early, most importantly, any unusual events that might signal human to human transmission of these viruses. Again, this is not the case now, but if it happened, we would want to be able to detect that occurrence as quickly as possible so that we can respond appropriately. So we want to make sure we're monitoring the viruses, understanding the differences between those in humans and those in animals, so we can inform our control measures, and also to describe what the type of illnesses and what are the risk factors for human cases as they occur. And again, it's this timely detection of these viruses in humans that may emerge and spread and cause illness in humans, and it requires multiple different surveillance approaches and systems to work together. And I would just wanted to highlight it on this side, some examples of different surveillance approaches that are used or can be used in countries for the rapid and early detection of cases. So I think what we're most familiar with is health facility event-based surveillance. So workers, healthcare workers being alert for the possibility of infections, knowing what the possible signs and symptoms of a zoonotic influenza virus infection might be. It's important that healthcare workers know how to respond, who they should report this, how they collect samples, etc. Community event-based surveillance can play a role. And there are laboratory networks, and there's surveillance in special populations. Media event-based surveillance also plays a role. We have our syndromic surveillance systems that are well established in many countries that we can use to look at any unusual signals that might indicate something different is going on. And we can use investigations and studies to get more information and dig a bit deeper into outbreaks, etc. So as I said, investigations are important. If there's any suspected or confirmed cases, it's really important to thoroughly investigate these and implement immediate public health actions. So this would include testing for animal influenza viruses in suspected human cases as part of the investigation and your laboratory protocols. In suspected human cases, asking for if persons had exposure to animals or travel history to areas where there may be outbreaks in animals, doing contact tracing and actively searching for other cases. And again, the important part of looking for any unusual events that might signal that the virus was transmitting from one case to another person. So not via an animal, but the virus was actually going from one person to another. And that would be an unusual event that would need to be followed up very, very closely. It might signal a change in the virus, and it's important to put in measures to mitigate and stop that transmission. The other important part is that in part of the investigation, it's important that public health and the animal health authorities work together to share information and do the investigation and the risk assessment. On influenza, it's very important the diagnosis has to be in the laboratory. So it's really important that there's appropriate specimen collection, and this is done in a laboratory that's qualified, and also that the virus, if there is one, that this is shared with with specialized laboratories for further characterization. Clinical management, an important aspect. Again, cases should be triaged, samples collected and treated appropriately. And I just wanted to highlight here there are clinical practice guidelines for influenza virus infection updated by WHO, and those are being updated and should come out soon. And they include the clinical management of zoonotic influenza infections. There's a lot of recommendations on reducing exposure. Basically it means, trying to limit the exposure of humans to the virus, whether in the environment or in animals or animal products, and protecting workers with personal protective equipment. And risk communication, very important. Having strategies to communicate risk to different groups, building trust, communicating as together with animal health authorities in a one voice approach, very important as part of the response as well. So I'll just conclude here, just to say that the summary of the current situation, there's been around 900 detections of H5N1 in humans, reported from around 20 countries since 2003. Again, these are cases where the person has had exposure to infected animals or environments where the virus may be. The infections can be severe and even fatal. We do continue to monitor closely all the animal influenza viruses, but especially those of this clade H5 viruses that continue to diversify genetically and spread geographically. It's a concerning evolving situation, but not an influenza pandemic. It's important that there is awareness and there are investigations if there are any suspect events, that we can identify events early on so that we can implement the right measures. And again, it's important that the global collaboration and coordination, both with other sectors, including the animal health sector, and with countries around the world. And again, we have a global influenza surveillance system that is the foundation for this global work. So I'll stop there and thank you, and look forward to answering questions later. Thank you.
Well, thank you so much, Dr. Hammond. That was really, really terrific. So we're now going to hear from our second speaker, Dr. Vivian Dugan, who's the director of the Influenza Division at the National Center for Immunization and Respiratory Diseases at the U.S. Centers for Disease Control and Prevention. Dr. Dugan is an expert in animal infectious diseases, and she provides programmatic leadership and overall scientific and administrative management of the Influenza Division's activities and functions. She oversees the synchronization of epidemiologic and laboratory science with informatics, and coordinates innovative intramural and extramural projects to advance the division's mission. So, Dr. Dugan, let's go ahead and hear from you, and then we will, I think, have some time for some interesting questions at the end. Please go ahead. Thank you.
Great. Thank you so much. Thank you, Dr. Dustman. And thank you very much to the organizers of ICAP, the Grand Rounds webinar, for asking me to speak today about H5N1, avian influenza. And of course, we just heard a really nice talk from Dr. Hammond, showing a fantastic overview that sets the stage to kind of talking into the U.S. H5N1 avian influenza situation, the current outbreak that's been occurring for about 5 to 6 months in the U.S. So what I'm showing here dovetails and is similar to some of the data that Dr. Hammond provided, some of the background of H5N1 from a global circulation perspective. So picking up from where she ended, she referenced 900 human cases of H5N1 globally, and these are shown on the graph on the screen. And so as you can see, in 2015, there were hundreds of cases, human cases of H5N1 in Egypt. So there's a lot that we do know about this virus in humans. It's not that new in many ways, but in some ways it is still relatively new to us as it's moved into a new host, which are dairy cows in the US. I'll talk about that in a second. So it's important to note that as we talk more about the current situation in the United States, the mortality overall, when you look at the evolution of H5N1 avian influenza viruses across over 20 to 25 years, and those 900 cases, the overall mortality rate taken together is a little bit more than about 50%, looking at all those cases since 1997. So historically, the human infections of H5N1, shown on the previous graph, have been really the result of poultry exposures from direct or close contact with sick or dead poultry, or visiting a live poultry market. And so this is really the theme and the general activity that has been documented again over several decades. Additionally, exposure to other infected animals such as swans and then now most recently dairy cows have also been that source of human exposure overall to H5N1 avian flu. We have seen, again, historically limited non-sustained human-to-human transmission in other countries. Generally, these are family members that are caring for sick individuals, sick loved ones, but we have not seen human-to-human transmission of H5N1 in the U.S. And this graphic just shows some of the different ways that that bird flu or avian flu from poultry can spread to people. So a little bit about the current situation in dairy cows in the U.S. I'm going to provide a little bit of an update and then not only what we're doing from a government, U.S. government response, but also outbreak perspective to the situation in cows in the U.S. So there is currently a coordinated U.S. government response to this outbreak of high pathogenic avian influenza in dairy cows in the U.S. And CDC serves as the lead for the human health aspect of that response. CDC is working very closely to coordinate, again, a One Health approach with the United States Department of Agriculture, or USDA, as well as the U.S. Food and Drug Administration, or FDA, and other federal agencies, where USDA is the lead on the animal side and the agricultural side of this response. And so this slide just shows some of our high-level priorities and what we're focusing on from a CDC perspective. Again, supporting and engaging our public health and agricultural partners in our states at our local and state and federal level, protecting human health and safety by really helping our state partners to stand up monitoring people who have been exposed, testing programs for anybody who has actually developed symptoms, and those individuals for, again, working with infected cattle and other animals, and really assuring that all of our state public health partners have the information and resources that they need to prevent human infections. So we understand that the risk to people to H5N1, we're trying to find more information through epidemiologic studies and also identifying ways to really prevent additional cases. We're also assessing H5N1 viruses in people and animals, again, dovetailing and complementing what Dr. Hammond talked about. We're looking for genetic changes in the H5N1 viruses that are circulating in animals, not only in the US, but globally. And then also any physical or phenotypic changes when we characterize these viruses in the lab that might indicate that this virus may be adapting or changing to spread more easily or more efficiently to and among people. So the current H5 situation in dairy herds in the US, so as of July 22nd, USDA has confirmed there is high path avian flu H5N1 in dairy herds on more than 190 farms in 13 states. That's shown on this map. All of these data are public. USDA, our agricultural colleagues, provide these data and they update them regularly. And so these illnesses in dairy cows were first observed in early 2024. And the dairy cows showed significant decreases in quantity and quality of milk. On March 25th, 2024, USDA reported that high-path avian flu H5N1 had been confirmed in cows and milk specimens from Texas and Kansas, with subsequent detections that occurred in states shown on the map. And again, I'll have the link there in case you want to go through and find more updates. So moving into the human cases, so this is really CDC's purview here. From 2022 through today in the US, there have been 14 human cases of high-path avian influenza H5 in people in the US. 13 of these cases in the US have occurred since March of 2024. There have been, so out of those 13, 4 of those human cases in 2024 have been among dairy workers that were exposed to, again, infected dairy cows. And these cases were reported in 3 states, Texas, Michigan, and Colorado. Additionally, 10 cases, again, looking at that total denominator of 14 over all history and time in the US, 10 of those cases have been associated with poultry exposures. So 9 of those 10 occurred in Colorado just this year in July among workers that were depopulating infected poultry that had H5N1. So the Colorado State Department of Public Health led the investigation on these cases that occurred in 3 different poultry farms. And from a CDC federal perspective, we provided field staff to support this investigation at the request of the state. The team included epidemiologists, veterinarians, human clinicians, and an industrial hygienist. And so we accompanied our Colorado health officials during daily visits to the affected farms to, again, monitor workers for any symptoms after exposure, test any symptomatic people, and of course, dispense antiviral treatments. And then, of course, follow up with people who have tested positive to collect additional information and assess the use of personal protective equipment and make sure that everybody was resolving their infection. So very, very fortunately, all of these human cases have been very clinically mild with no hospitalizations. I'll talk a little bit about that in a second. So a little bit about the genetic aspect of the H5N1 virus in the U.S. from what we're seeing in dairy cows. Any specimens that are received and from people in the U.S. are sent to the U.S. CDC, and of course we are characterizing those specimens to really get as much information as we can about the virus itself. And so we do a lot of genetic sequencing of the virus from both human cases, again, where feasible, and of course USDA is providing a lot of data from the animal side. And so when we look at all of these data together, we look at, again, the clade of this particular H5N1 virus. They are that same clade that Dr. Hammond mentioned, 2344B, for all these cases. And this is the clade that's widely circulating, not only in wild birds and poultry, but also now moving into people here and spilling over. It's important to note that the available information currently indicates that when we look at the sequence of the viruses from people, that our current diagnostics for H5 will be able to continue to detect this virus, so it's not changing, that there are no known markers of influenza antiviral resistance that would impact our medications. And then also the virus is very closely related to candidate vaccine viruses that we work in support of World Health Organization that could be used to make a vaccine if that were needed. A little bit about symptom monitoring. And so CDC works very closely in collaboration, again, with our state and local health departments to monitor people that may have been exposed to H5N1 for any signs or symptoms of illness. And so we recommend that people who have that direct or close exposure to infected animals should be monitored for illness while their exposure is ongoing and for up to 10 days after their last exposure to that infected animal. Some of the signs and symptoms may include fever, feverish, having a fever, a cough, a sore throat, runny or stuffy nose, again, influenza-like illness, any muscle or body aches, headaches, fatigue, eye redness, or conjunctivitis, which has been a very strong symptom that we've been seeing here in the U.S. And then again, of course, something more serious is shortness of breath or difficulty breathing. Less commonly are diarrhea, nausea, vomiting, or seizures. And so we've been working very closely again with our partner organizations and clinicians to really stress the importance that if signs or symptoms develop in people who have that exposure or who may have been exposed to an infected animal, people need to seek medical care and be tested for influenza as soon as possible. And so our state and local departments, health departments, are monitoring workers on the impacted farms and are facilitating testing and treatment. For our ongoing monitoring efforts, again, these are people who are working on dairy farms as well as poultry farms. We've, for the cows itself in the dairy space, more than 4,500 people have been monitored as a result of their exposure to infected or potentially infected animals. And at least 230 people have developed flu-like symptoms. They've been tested as part of this more targeted situation-specific testing. This testing of exposed individuals or people who do develop symptoms is happening at the state and local level, and CDC conducts that confirmatory testing. That's how we get those specimens into our labs here in Atlanta, Georgia. And so then as part of the outbreak investigation and monitoring for Colorado itself, more than 600 workers performing depopulation activities for poultry on those 3 farms were screened for symptoms with, again, those 9 individuals testing positive for H5. So that's what's happening in our targeted surveillance and support for monitoring those who have that direct exposure. But when we look at our overall U.S. surveillance system, we maintain a very comprehensive layered system that looks at all flu all the time. So it's just seasonal viruses. It's not just seasonal. It's novel influenza, so swine influenza, avian influenza. And this is a collaborative effort that we work again through our partners. And so our surveillance data are collected from a variety of sources to capture influenza activity whenever and wherever it's happening. It doesn't stop. It persists throughout the summer months here when seasonal activity is often low. And so these data are collected again from our partners and vital statistics offices, healthcare providers, hospitals, clinics, and emergency departments. And the data are reviewed comprehensively each week. And so we are been actively looking and monitoring and analyzing these data and other systems to really make sure that we are not seeing any concerning changes in any of these, again, national systems for that would suggest that H5N1 may be spreading among people. And so as part of our regular public health partner testing, more than 43,000 specimens have been tested since March of 2024 using a protocol that would find a novel influenza virus. No additional cases have been identified through this national system. And again, no indicators of unusual influenza activity have been determined. And then again, ongoing efforts here really providing some recent results from a seroprevalence study that was led by our Michigan State Department of Health partners, where we provided technical support. So in June of this year, blood specimens were collected from 35 people who work on dairy farms in Michigan with infected herds. And so study participants were from a bunch of different counties, and they had different roles on these different farms, but most of them worked with sick cows directly, and fewer than half of them reported using masks or gloves as far as PPE. So we tested these specimens for antibodies against H5N1, as well as seasonal influenza viruses. And so what we found is that none of these participants out of the 35 people showed neutralizing antibodies for H5N1. And so these data are consistent with other data demonstrating the seroprevalence of H5N1, even among workers with known exposures. We know that's quite low. And this also supports the concept. Again, the numbers are small here, but we're starting to get preliminary data here. This supports the idea that asymptomatic infections in people not actually causing great disease or providing any additional routes of infection at this point. So we're doing more in this space. We're trying to really make sure that we are supporting our states to understand what the risk may be. Is asymptomatic infection occurring? The second phase of the study is ongoing, and we're also working with our Colorado Department of Public Health partners to do a similar study as well. And then quickly, I just want to talk about, again, similar to the risk assessment tool that WHO has, we also have a similar tool that we use for basically planning from a government perspective, from a preparedness perspective. And so this is our influenza risk assessment tool. This is all about pandemic influenza and preparing for that. So this is a tool that was conceived about 10 years ago with CDC and other US government partners to really assess the potential pandemic risk posed by influenza A viruses that are in animals, but are not circulating in people. The tool itself does not predict a pandemic. It's not used to assess the current risk to people. It's really, again, from a planning and programmatic perspective, as we have many of these viruses that spill into people at times. And so we use the tool and we run the tool when there's a new zoonotic virus that may be circulating that's different from what we've seen before. And so these are the risk elements that we use to assess the virus. And so subject matter experts are participating in this, again, using data that are available to understand the risk. And so again, it answers two questions. If this virus were to emerge in people, what are the chances that could happen? And then also, what is the impact to people should that virus emerge to sustainably transmit person to person? And so these are our results. I won't go through them all the way, but I will just say that the 3 viruses that are all from the same clade, H5N1, are shown here with the arrows, 3 consecutive years. And so the risk currently from a pandemic perspective as a potential pandemic virus is that this is a moderate risk to be a potential pandemic virus. And so this updated information includes everything that we know up until end of June of 2024, and the report is on our website. But overall, I think this is not surprising. I think we're in a very good preparedness posture, and so I'll talk a little bit more about that next. So overall, the public health risk, when we think about the US and where we are right now, where these H5N1 viruses are circulating, the overall risk to the general public here in the US remains quite low. However, those who have that recreational exposure or occupational exposure to infected animals may be at that increased risk for infection and should be following recommended precautions. So of course, monitoring for any symptoms and first exposure for at least 10 days after that last exposure, testing should they develop any kind of symptoms, including conjunctivitis. And then also we have updated guidance on our website, and happy to share that link as well. And so when we think about where we are from a pandemic preparedness perspective, we are assessing this virus as much as we have specimens, understanding that a lot of data is coming from the animal perspective, and making sure that the virus is not changing in any way that would impact our current countermeasures, candidate vaccine viruses, antivirals, and diagnostics, and making sure that we're getting these data out into the public as quickly as we can. So these are some of the resources from CDC, but thinking about a potential pandemic, which is part of our mission here at CDC, this again remains a moderate potential pandemic virus at this point. We know that can change quickly, but as we get more information, we continue to update this and get these data out so that we can really assess this from a global perspective. So additionally, we have a wide range of resources here, but I encourage you to take a look at some of these resources for what we're doing in this space. And for that, just want to thank you. And thanks for allowing me to share this information, and happy to take any questions.
Well, that was really just terrific. So thank you very much, Dr. Dugan. Really thanks to both of you. So we do have some questions in the Q&A box. And I may just take the lead as moderator and ask you, Dr. Dugan, to tell the audience a little bit more about what does it mean when you say moderate risk, that we're, at a point where you and others feel that the pandemic potential risk in terms of illness involving humans for H5N1 is moderate. I'd love to hear more about what exactly that means.
Sure. So when we developed the risk assessment tool, it's really based on a scoring metric. And so 0 to 3.9 is low, moderate is 4 to, I think it's 6.9, and then 7 to 10 is really that high. So that high risk. And so the elements that go into the IRAT are looking at, there's 10 major areas. So looking at the genetic changes of the circulating viruses, looking at whether or not these H5 avian flu viruses have the ability to change how they bind to host cells. That's a really host-specific important piece of this. And you probably are very familiar with this with SARS-CoV-2 and the spike protein. So the HA protein is how the virus binds to cells. And so right now, the H5N1 viruses are very avian in their receptor binding. They are not mammalian in their receptor binding. So they really lack the ability to bind a mammalian cell. But also, I mean, there's a lot of other aspects that go into this, looking at transmission in laboratory animals, looking at antivirals and treatment options, population immunity, looking at disease severity and pathogenesis, what we know so far, looking at the genetic and antigenic changes to vaccine candidates. Do we have a vaccine candidate? Looking at the global geographic distribution, as well as the infection status in animals, and then human infections and transmission. So all of those different areas kind of go in together to generate those scores by subject matter experts and get into that moderate risk space.
Okay.
Does that help kind of explain the little bit about what goes into this?
It certainly explains what goes into it. I'll follow up with one more, and then we have a bunch of questions I want to get to from the Q&A, and that is just thinking about the coming flu season. and do our, influenza diagnostic tests, do they adequately distinguish H5N1 from other kinds of influenza? Are we going to know, or is it going to require specialized testing? And is there anything people should do as we think about the flu season approaching?
Sure. So a little bit about that. All of the public health labs in the US have an H5 specific assay. It's part of an algorithm that starts off with flu A, B, is it seasonal? If it's not seasonal, is something new? Then we reflex to H5, of course, if there are epidemiologic criteria that would support that. So that's one space. And so to give you an example of how the system works, we've detected 7 variant influenza cases or swine influenza viruses that have spilled over into people just from using this system from a national perspective alone. So we do have an H5 specific test. Another thing that we're doing in the kind of commercial space is that we are working to fund commercial diagnostic companies in the US from a preparedness perspective only to have an H5 assay that's very specific that could be rolled out from a high throughput perspective. So I think we're doing both. We have a lot of this in the public health space and also in the global space, an H5 test that can be used. But from a commercial space, we are moving that forward again to be really ready in the US in case the situation changes.
Great. Okay, so let me select some questions from our open Q&A. And perhaps this is a question that's good for Dr. Hammond as well as Dr. Dugan. Are there particular thresholds that you would use to define the situation as a pandemic? And I certainly know WHO has experience in thinking about this question with SARS-CoV-2 and other infections, but I think it'd be just great to hear about it. So maybe Dr. Hammond, do you want to take a stab at that first?
I can start, and Vivian can complement. But yeah, thanks for the question. I think it's really important. I think I mentioned a bit, it takes a virus that's different from the seasonal influenza viruses. So it would be something different, something, it's an animal virus, or that's a novel, reassortant virus, and that the population has very little immunity to, so that infections can happen quite easily. And most importantly, that the virus is not just adapted to circulating in animals, but that it acquires that capability to transmit from one infected person to another. So that, respiratory transmission from person to person is very important. So that would change quite a lot. And it, and of course, there's many aspects that would come into the considerations for calling this a public health emergency of international concern, possibly leading to a pandemic, but the severity and how widespread it is and how likely it is to be sort of minimized from spreading from just a localized level to more broadly. So, but really what we look at are, the, virologic changes, but also the changes in the epidemiology.
Maybe if it's okay, Dr. Dugan, I'm going to move to a different question. We had somebody ask about wastewater surveillance and whether that is complementing the surveillance approaches, certainly in the dairy farm areas. Is that something that's in use?
Yeah, great question. So we recently rolled out influenza A, again, kind of family level wastewater surveillance that had been happening for quite some time, and to really understand the utility in this situation. But that's still ongoing. It's really more for seasonal influenza, but we have it. And so now we're moving into the H5 space. And so our public health partners and outside partners from CDC are actually running H5 specific wastewater testing across the US. And so what we're finding is that it's really good at flagging animals being positive for H5 so far. But again, we're still really applying that, and we hope to have more data on our website. It's up there now to understand how does that complement all of our existing surveillance systems for looking for any early signals.
Great. And maybe on the surveillance side, I was going to ask Dr. Hammond, do you feel that the sort of global network of surveillance is sufficient? Are there areas or are there gaps where you're concerned that there is infection and transmission going on that we don't know about? What's your thought? What is your thought on that?
Yeah, I think that's a good question. And sometimes when we look at the map of where H5N1 infection in humans have been detected, there seems to be areas where there have been none. And the question is, is it true, or is there a gap in surveillance? And it's probably both. I think, we have a couple of gaps that can be improved. I think one is on the animal surveillance side. So it's important that that we have good animal surveillance, so we know where these viruses are circulating in animals, and so we can target better our surveillance for infections in humans. So I think there are some gaps in animal surveillance. There are gaps in influenza surveillance in some areas, and WHO works very hard with partners to improve and enhance surveillance for influenza and other respiratory viruses in countries around the world, so that we can be on top of these things. I think that as Dr. Dugan said, there was some serologic evidence that it seems that in the US, at least, in certain situations, there seems to be few asymptomatic infections, and there have been serology studies elsewhere in the world finding similar things, that it doesn't seem that we're missing on a broad scale infections, but we continue to make sure that all the national influenza centers are prepared in terms of having the diagnostics to test. And it's really important that as you were talking about the testing, if there's any the lab should be able to test for influenza A viruses, and be able to subtype a virus that'll not subbed up and sent to a collaborating center, because those are the areas where we need to be jump on and make sure that there are no unusual viruses circulating. So in terms of a global system for doing that, for checking the circulating influenza viruses in people and having a system for further characterization, I think that the system works well in that regard. But there are certainly some areas where surveillance is not comprehensive or representative as it can be. But it's understandable. There's a lot of, in the world, there's a lot of diseases, and there's a lot of other competing pathogens and priorities. So, it's not an easy thing to fix. Over.
Wafa or Joey, any questions that you'd like to raise before we wrap up? And if not, there's still some questions in the Q&A I can move to, but I wanted to just give you both a chance.
No, why don't you go ahead, Jessica? I was thank you, both of you, for the presentation. I was wondering about, there's always the fear of misinformation, disinformation, as we've learned from COVID-19, and I'm sure that there's now kind of both the risk of such misinformation, both in the context of potential for human disease, but also what's happening in the animal sector and the safety of the milk supply and so on. So I'm just wondering if you want to touch on that.
I can try. I think from a CDC emergency perspective, we are trying to really follow those rules that Aspen laid out for emergency and risk communication. So being fast, being right, and saying what we don't know, and really being honest and forthcoming, as sharing what we know when we know it, and trying to get that message out and make it as public as we can. And so this webinar is one great way to do that. To again increase that awareness and making sure that people know where they can go to find more information. So I think we're kind of doubling down on that effort and making sure we're using social media, all the different tools that are available to us, again, to be fast, to be right, and to be honest about what we know and what we don't know, but also really working with trusted messengers to get that message out. We know that sometimes the message doesn't come from a great place of trust, sometimes when it comes from federal and public health officials, and so using those trusted partners to share messages.
Sounds great. There is, maybe this is a good quick question, a question in the chat. wondering about the incubation period for H5N1. Is that something that you know off the top of your head? And if you don't, that's fine. I know that's.
Yeah, I would say it ranges for all the H5s. There's quite a diverse range. But what we can do is we can point you towards our technical report where we kind of summarize all the H5 all the time. We try to update this every quarter or sooner. But I think it's similar in many ways to seasonal flu, where it can happen pretty quickly if the virus starts to replicate, but there's certainly it can be longer than a couple of days at a time.
Great. Okay, well, I think unfortunately we have to wrap up. I think there's lots of, additional questions. We could probably have another, 15 minutes of conversation. The webinar recording and the slides will be posted on the ICAP website, and a few of you were asking about that, so that resource will be. So just in closing, I really want to thank Dr. Hammond and Dr. Dugan for these just fantastic updates and the conversation that we've been able to have. It's really very wonderful. So thank you, everybody, and take care.
Thank you. Bye-bye.
Okay.